Hello, everyone. It's highly welcome to see a lot of you here. And so it's my great pleasure to introduce Professor Huimin Zhao. And he is from the University of Illinois at Urbana and Champaign. And he's the Stephen Miller Chair Professor of Chemical and Biomolecular Engineering at UIUC. And also the director of the NSF AI Institute for Molecular Synthesis. And he received his bachelor's degree in the University of Science and Technology of China in 1992, right? And then he came to the Caltech and for his PhD with the Nobel laureate Francis Arnold, and in the topic of direct evolution. And this is something that he's going to talk about today. And before joining the UIUC in 2000, and he's the project leader in dog chemistry as for the industry operations. And he joined the UIUC in 2000, and then 8 years later he was promoted into the full professor. And Dr. Zhao has authorized and co-authorized over about 400 papers, and also issued about 30 patents. Some of them has been already licensed to the industry. And he give a lot of plenary talk and the keynote talk and invite talk, and all over the world. And what is more important to mention that, and he has supervised more than 30 PhD students, and also the postdoc, which has been already become a professor and over worldwide, for example, in the United States, in China, Korea, and Singapore, and so on. And besides this, Professor Zhao has also received numerous research and teaching awards, and including, for example, the ECI, the Enzyme Engineering Award, and also a Marvin Johnson Award, and so on. And his topic today is really interdisciplinary, which is cover the biology and chemistry, and also the machine learning, data science, and also engineering. So this is something that we really appreciate that this our institute is something about interdisciplinary. This is our focus. So you are actually the right person. So actually, we approached the Professor Zhao a number of years before, and we should have you here in 2020, right? Yeah. But due to the pandemic, it was delayed and a lot of times. So it actually give us a lot of curiosity to see. Okay, so let's give the hand to Professor Zhao, and we are looking forward to your talk. Thank you.
Okay. Yeah, nice. Thanks for your nice introduction. Indeed, I was supposed to be here actually in June 2020, right? But I finally made it after a few attempts. It's a great pleasure to be here. This is my first time actually to visit this university, but I've been to Denmark a few times. And today I will mainly talk about a topic dear and near to my heart. It's called direct evolution. So direct evolution. Oh, no, I haven't turned on. It's a very actually simple approach for engineering biological system. So it is basically is to introduce the mutations into the targeted gene pathway or the whole genomes, and then screen the library using high throughput screening or selection method to identify the functionally improved variants, and then we actually will repeat this cycle many times, right, to obtain the desired function. And although this approach is very simple, but actually it's very powerful. It has been used by many groups around the world. And the 2018 Nobel Prize in Chemistry actually was given to the 3 pioneers in this area, including Francis Arnold, who was my former PhD advisor. And actually, my PhD thesis actually is entirely focused on direct evolution. I joined her lab in 1992, and then left her lab in actually 1998. So this actually is the photo of the cover of my PhD thesis, right? So I developed a number of those direct evolution methods, actually that really laid the foundation for this whole field. And direct evolution is not just actually for protein engineering. Actually, it also can be used for engineering of DNA, pathways, or even the whole organisms, right? For many different applications. And the field actually really started, I think, in the early 90s. Right? Here I listed those kind of milestones in this field. And the ones highlighted in red are related to protein engineering, and the ones highlighted in green are related to pathway engineering, and then the blue ones are related to the genome engineering. As you can see, the development of this whole field. And in recent years, more and more efforts actually were focused on the genome engineering using direct evolution approaches, and also the development of those in vivo continuous evolution strategies. Right? So you can actually repeat the direct evolution cycle autonomously for many, many times. And of course, direct evolution is also not just for engineering of biological systems. It can even be used to evolve processes. This is actually an abstract published in a real journal. It's the Current Biology, I think, in the 1990s. And it's very interesting, so that's why I actually copied it and pasted it here. So it says that the success in academia is hypothesized to require specific phenotypes. And in order to understand how such unusual traits arise, we use the human clones to identify the molecular events that occur during the transition from graduate student to professor. And a pool of graduate student clones was subjected to several rounds of random mutagenesis, followed by selection on minimal money media in the absence of dental insurance. And the students surviving this selection were further screened for the ability to work for long hours with the vending machines, snacks, as the sole carbon source. And the clones satisfying these requirements were dubbed postdocs. And in order to identify assistant professors from amongst the postdocs, this pool was further mutagenized and screened for the ability to turn esoteric results into a 50-minute seminar. That's exactly what I'm doing now. And finally, these assistant professors were evaluated for their potential to become full professors in two ways. First, they were screened for overproduction and surface display of stress proteins, such as heat shock protein 70. And assistant professors that displayed such proteins, so-called stressed out mutants, were then fused to the M13 coated protein displayed on phages and passed over friend and family members column to identify those that were incapable of functional interactions. And these were called full professors. Although these mutants arose independently, they shared striking phenotypes. They included the propensity to talk incessantly about their own research, the inability to judge accurately the time required to complete bench work, and the belief that all of these ideas consider good thesis projects. And the linkage of all these traits suggests that these phenotypes are coordinately regulated, and preliminary results have identified a putative global regulator. And studies are currently being conducted to determine whether overexpression of this gene product in postdocs and graduate students can speed up the graduate student to full professor evolution process. Right? So, and indeed here actually mentioned the two actual work that are got a Nobel Prize. One is phage display, right? And the other one is the direct evolution, right? These are all related. Okay, but even for myself, right, I also evolved my own career. So this actually is the first time I actually, as a graduate student, to present my scientific work in a conference, which was held in San Diego. And then I graduated in 1998, and then actually I even went back to Caltech, with my family, and then actually 4 years ago I was also attended the Nobel Prize ceremony in Stockholm. But even look at the Frances, she also kind of evolved, at least her hair color changed over the years. But anyway, so my lab is really focused on direct evolution, right? But now we are really working at the interface of synthetic biology, machine learning, and laboratory automation. So we are very interested in developing foundational tools for synthetic biology applications, particularly in the form of a Biofoundry, which is a kind of automated robotic system. And then we actually also develop various tools for protein engineering and pathway engineering and metabolic engineering, right? And then we try to leverage this technology platform for three main applications. One application is related to the discovery of natural products. And also we try to characterize their important biological activities and biosynthetic mechanisms. And then we also are interested in leveraging the synthetic biology platform for the engineering of microbial cell factories for production of chemicals and materials. And then lastly, we're also applying the synthetic biology tools, particularly the genome editing tools, for the engineering of mammalian cells for gene therapy applications, and also the fundamental studies of biology, like the chromatin structure and dynamics. And clearly, there are a lot of things that we can do, but there is one unifying theme, which actually is the direct evolution. We actually use the direct evolution for many of the projects that we're working on. And so I want to talk about a few examples how we can really apply direct evolution for engineering of proteins, pathways, and the whole genomes. So the first example was done actually many years ago. We basically tried to apply direct evolution to create new protein functions. Right? And actually, this actually is a pretty challenging work. Direct evolution actually has been very successful in improving the existing protein functions, right, through the iterative cycle of diversity generation and the screening or selection, because we can accumulate those beneficial mutations through multiple runs. While for creation of novel functions, it is challenging mainly because it may require many simultaneous mutations, right? And that's very hard to actually identify those mutations. And because you can actually just do a simple calculation. A typical protein is about 300 amino acids. And if you create a single mutations, the library size is about 6,000. If you want to create two mutations simultaneously, the library size is already 18 million, right? And if you create a triple mutations simultaneously, the library size already is bigger than any of the screening or selection method that we have, right? So basically, the bottom line that you cannot find those simultaneous mutations through the library screening approach. And so that's why we came up with an approach called the in vitro co-evolution strategy. So our idea is to actually create some intermediate functions that can serve as the bridge between the wild type of function and the novel function. So as I shown here, in the sequence space, the wild type of function is here, and then this novel function. And then we try to actually create some intermediate function, right? And then evolve them, using the direct evolution approach, gradually. And then we just continue this process until we actually kind of obtain the novel function. And to demonstrate this simple concept, we actually use the ligand bind domain of the estrogen receptor. This receptor actually will bind the estradiol, and that is the native ligand, right? And then we use another substrate called corticosterol. And as you can see, these two substrates are very different. They differ at like 5 different positions, right? And then what we did is that we choose 2 intermediate ligands. One is the testosterone, one is the progesterone. And then the testosterone actually differ from the estradiol at 2 positions, right? These 2 positions. While the progesterone differs from the testosterone at also 2 positions. So then basically these 2 ligands serve as kind of the intermediates, right? That can bridge the evolution of the estradiol to corticosterol. And then when you actually do the dose response study, you will find that the estrogen receptor actually respond to the estradiol at a very low concentration, right? It's almost like less than a nanomolar, right? While it didn't show any activity or any response towards the corticosterol. And also it didn't show any activity towards the progesterone. So it only showed some activity towards the testosterone, right? But this is the intermediate function we want, right? So that we can use the direct evolution approach to improve it. So then what we did in the first round, we just create a library, random mutations library, and then screen the mutants for the increased response toward testosterone. And indeed, we got a few mutants showed improvement. And then we can repeat this cycle. And then some of the mutants also showed some activity towards progesterone, but no activity toward testosterone. And then we can actually repeat this cycle, right? And we found further improved mutants that have a higher response towards testosterone, and also showed a high response towards progesterone, but it still didn't have any activity towards corticosteroid. And then we can actually switch the substrate to progesterone, because that response is already pretty decent, right? And then we screen the library for the mutants with increased response toward progesterone, and indeed we obtained some mutants with a higher response, but it still didn't show any activity. And then we actually did another round, and now we obtained the 2 mutants actually showed a response towards corticosteroid, right? So basically through this co-evolution strategy, we actually create the new substrate specificity in the protein. And this approach actually also was used by scientists at Merck to actually engineer transaminase enzyme for the synthesis of this drug. This drug actually is used for treatment of diabetes. So what they did is that they actually used this co-evolution strategy to make this transaminase work towards their target substrate, and then they actually even commercialized this whole process, right? And then, as I just mentioned, creation of novel protein function indeed is very challenging, because it requires many mutations that are very hard to predict, right, or to identify. So then in the recent years, we actually came up another new strategy to create the new protein functions, in which we try to combine photocatalysis with enzyme catalysis. Because photocatalysis actually can impart new and unique reactivities to enzymes. The radicals are very reactive, right? They can react with many compounds. While enzymes, actually, they have limited reactivity, but they have a very high selectivity, because they have a protein scaffold, right? And the active sites are well positioned, right? And so what we try to do is really try to combine the advantage of photocatalysis with the advantage of enzymes to really create new enzymes that can have new reactivities. And in the past few years, we initially actually combined enzyme with photocatalysis just separately, right? But combined photocatalyst and enzymes together and in one reaction vessel, and we try to explore the synergy, right? Basically, want to make them work, cooperatively to produce some chiral compounds. And then later we thought, why not we actually directly engineer the enzymes with new reactivity? And so we actually use the light to basically activate the enzyme to create new radicals, like the FAM radicals. And then we actually, the radical will catalyze basically the chemical reaction that cannot be easily catalyzed by chemical catalyst. And clearly they cannot be catalyzed by enzymes, right? Because the enzymes don't have that activity at all. But even for that chemical reaction, actually it's very hard to achieve by chemical catalyst with high selectivity. So today I actually want to use a recent example to highlight this strategy. And in the particular reaction, we are interested in this radical addition to alkene reaction, right? And this example is the one I just mentioned, right? We actually did the electrophilic radical addition to the alkenes. And this coupling, CC bond formation, actually is very challenging. Mainly because the product actually will be like a racemic, right? It's not like an enantioselective pure. And so we were wondering whether it is possible to create an enzyme that can catalyze this reaction selectively. And this reaction actually has been done by some chemists, which was done actually more than 20 years ago by a group in Japan. And they actually used a photocatalyst here. This is kind of similar to the cofactor in the enzyme that we will use. And this reaction can happen in vitro, but as you can see, they produce a number of products, right? And those products are also not enantially pure. So then what we thought is that possible we actually use the enzyme, right, to catalyze this CC bond coupling reaction in an selective, enantioselective manner. So we basically use the light to generate the radical in the cofactor, the nicotinamide, right? And then we actually added the substrate, but then this radical actually will, not the radical, the hydrogen atom on the nicotinamide cofactor will be transferred to this conjugated product in a selective manner, because it only can actually attack in one direction. And then we can achieve the enantioselectivity we want. So then what we did is that initially we actually don't know which enzyme will work, right? So we actually bought a number of those commercial keto reductases. And we screened more than 24 those enzymes and found a few of them actually showed some activity towards this reaction. But the selectivity is not very good. So we actually selected one of them as the starting point for further direct evolution studies. So we identified the residues that interact with the substrate and then did a saturation metagenesis. And then we use HPLC to basically analyze the product that catalyzed by those mutants. And we found a few of them actually showed improved selectivity. And then we use those improved variants as the starting point for the next round of improvement. So we just repeat this cycle, right, to introduce more mutations in the enzyme. And we did 3 rounds and accumulated 3 mutations. And then the triple mutant actually showed a really high enantioselectivity towards the 2 substrates that we use. And then we also evaluate the catalyst, whether they can be used for synthesis of other molecules. It's not just the substrate that we use for the engineering, right? So, and indeed, the engineered enzyme also showed a pretty good activity towards many different substrates, right? And this actually is important for chemical synthesis, right? We want a catalyst that will work for many different compounds. So this is actually an example for protein engineering, particularly is to create new functions. And then we also want to actually use the direct evolution approaches to engineer the whole organisms. And so my vision actually is to use a genome-scale engineering tools like CRISPR or RNA interference, etc., to perturb the expression of the genes in the organism, all the genes in the organism, and then try to use the high-throughput screening or selection method to identify the improved variants, and then we can accumulate those beneficial mutations, right, through multiple rounds. So basically, we're performing direct evolution of an enzyme on the genome scale, right? So the concept is the exact same, but now we're basically dealing with many genes instead of just a single gene. And in metabolic engineering, actually, the traditional approach is the rational design approach. Basically, is to apply omics tools like the metabolomics tools or the RNA-seq, etc., to analyze the organism, try to identify those rate-limiting steps, and then use the genetic engineering tools to either overexpress genes or downregulate genes or knock out the genes, right? And then they have to repeat this cycle many, many times as well, really try to further improve the production of the targeted compound. And another approach is the so-called adaptive evolution. So basically just do the serial culture transfer. And that approach actually is very powerful, but it's only limited to the phenotypes that are related to cell survival and growth, right? And that actually really limits its application. So in the past 10 years, we try to develop those genome-scale engineering tools that will enable us to introduce basically mutations everywhere or anywhere in the chromosome or in the genome, right? So because of time limitation, I will not talk about all these approaches. I only want to highlight one of them. Is the CRISPR-based approach for genome-scale knockout. And what we did is that we actually synthesized an oligo about 120 BP long, which contains 2 short homology arms, which is about 40 BP long each. And then in between the 2 homology arms, there's 8 BP deletion, right? And these 2 homology arms, of course, have the sequence that are identical to the target gene, right? And then for the 6,000 genes in Saccharomyces cerevisiae, we actually synthesized 24, those 24,000 those oligos. And the reason we have to synthesize 24,000 because we also have to include a guide RNA. And this guide RNA, we don't know which one works well, right? So for each targeted gene, we basically designed 4 guide RNA in order to make sure one of them will work well. So then we actually use the microarray to synthesize 24,000 those oligonucleotides, and then cloned those oligos into a plasmid that contains the Cas9 protein already. And then when we express this library into yeast, and then the guide RNA actually will take the Cas9 to the right position in the chromosome and then to introduce the double-strand break. And once the double-strand break is introduced, and then through homologous recombination, the 8 BP deletion actually will be introduced into the chromosome, right? And because it's 8 BP deletion, so there will be like an open reading frame shift, and then the protein will be inactivated. And then we actually screen the library, try to identify those functionally improved variants. And as a proof of concept, we only picked a phenotype that is also related to actually inhibitor tolerance. And that's because we can use the next generation sequencing to identify the clones that actually survived in the high concentration of the inhibitor very easily. If it's like a chemical, then we have to use some mass spec, right, to really identify. That's kind of more tedious. But in the beginning, we don't know whether this genome scale knockout strategy will work well or not. So we actually did a kind of a proof of concept study. In the presence of this drug, canaverin, the yeast cell actually will be killed because this is a potent drug. But if the transporter CAN1 is knocked out, then the drug cannot enter the cell anymore, right? So then actually the yeast cell can survive, can grow. So then what we did is that we transformed this library of genome scale knockout into yeast and then added the drug, canaverin, and we found a few of them indeed actually can survive. And then we sequenced them, sequenced just the plasmid, right? And we found that all this plasmid contains the guide RNA that target this CAN1 gene. And so this is a very encouraging result. It means that out of 6,000 possible the genes, right, we actually can identify the exact target that is responsible for the tolerance towards this canaverin. Right. And then we actually used this library, this strategy to improve the tolerance of the yeast towards the 2 important inhibitors, the acetic acid and the furfural. Both are present in the lignin cellulosic hydrolysis for like a biochemical or biofuel production. And in the first round, we actually identified some targets that indeed actually improved tolerance of the organism towards furfural and also the acetic acid. And we actually know the exact target. And both targets actually are involved in post-trans protein. The students want to graduate. They just want to get some data and then leave, right? So that actually is more like for genome engineering. But in all these examples I showed, right, it requires the iterative cycle of direct evolution And it's still, it's pretty tedious because you have to do many runs, right? In most labs, they only did like a number of runs, right? That's fine. But if you really want to develop a product that can be commercialized, then I think you probably have to do many, many runs, right? Or 30 runs or even more, right? So then that becomes also a very time-consuming step. So that's why actually in the past few years, we also try to really develop the automation, right? And also AI tools to accelerate this direct evolution cycle. So we actually built a Biofoundry called the Illinois Biofoundry for Advanced Biomanufacturing, or iBioFab in short. So as shown in the video, basically we integrate more than actually now 40 instruments into a large platform. And in the center of the platform, there's a robotic arm that will take actually the 96-well plate and transfer it from one instrument to another. And there are many instruments on this platform that include like the PCR machine, the centrifuge, the incubators, the plate readers, the barcoders, and also, of course, the liquid handling system, right, or DNA fragment analyzers. So there are many instruments, more than 40 instruments, basically, they're all kind of integrated together, right? And we try to use this system for different applications, like protein engineering, genome engineering, metabolic engineering, or even just library screening, right? And those different workflows are very different. So our idea is really to break those workflows into kind of process modules. And those process modules will be further broken down into like unit operations. And these unit operations are more universal. They can actually work basically in different workflows. And then we have the operating system that it will control all the instrument. And then we also have a scheduling software basically we can use to program, right, the instruments to create the custom designed workflow for different applications. And so we were kind of inspired by the development of the computers. Many years ago, we actually built this iBioFab in 2014, almost more than 80 years ago, right? And for computer, actually, coincidentally, a group of faculty at our university built one of the first computer in 1952. And that computer consisted of like 2,800 vacuum tubes and weighed about 5 tons. So this is a photo of that computer, right? So of course now we all have iPhones or smartphones. That is actually a much better computer than those older computers. But the computing power of that computer already exceeded all the computers in the Bell Labs combined. Right? And for computers, of course, it went through many years of development. On our campus, the many generations of computers were developed. Currently, we have the Blue Waters on campus that actually really enables to do computing anywhere, anytime. And for the Biofoundry, we actually developed the second generation the system about 2 years ago. Basically, we incorporated more instruments, right, to address the limitations that we encountered in the past few years. And so I imagine that through many decades of research, eventually we also will have a Biofoundry that we can use to engineer any organisms for production of any molecules. And we are not alone. Actually, many universities are interested in building those Biofoundries. So actually, we formed a global Biofoundry alliance in 2019. It's before the pandemic. And currently, there are more than 30 members involved, and it's growing. Right? So in the consortium, basically, we try to share the knowledge and the tools that we developed and try to make the Biofoundry more accessible by the broader research community. So then in the past 8 years, my lab is really trying to demonstrate the unique capabilities of the Biofoundry, and in order to convince basically people to use the Biofoundry more and more, right? So the first demonstration we did is actually for protein engineering. So in that example, we try to synthesize TALENs, which are important genome editing tool. Although CRISPR is now commonly used, but TALENs still have some niche applications. And so in this project, we actually create a library of those repeats that will recognize like a nucleotide sequence, right? And then based on the target sequence, we can pick those repeats to create a custom designed DNA binding domain that will recognize that sequence. And then also fuse it with the nucleus domain, so we can create artificial nucleus called the TALEN, essentially. Right? So that TALEN will recognize whatever the target sequence and introduce the double-strand break. And then we can actually build this TALEN and transform it to E. coli, and then do the cell culture overnight, and then we can isolate the plasmid. So we develop a fully automated workflow, right, to basically synthesize TALENs in a high throughput manner. We can synthesize almost 400 TALENs in a day, and with very low cost. Right? So this really, helped, the development of this technology. Imagine if we don't have the TALEN nowadays. No, imagine if we don't have the CRISPRs nowadays. This will become very useful, right? Because everyone now will use this technology because they can just synthesize the TALENs. But unfortunately, the CRISPR just developed very shortly after the TALEN was invented. So now, of course, for CRISPR, you can just synthesize the guide RNA, so you can do it very quickly. While this one requires the assembly, right? And then actually, as I mentioned, the TALENs indeed have some niche applications. For example, if you're interested in treatment of mitochondrial disease, right? So you cannot actually deliver the CRISPR into the mitochondria, right? Because the guide RNA cannot enter the mitochondria. While for the TALEN, it is a protein. You just need to fuse the TALEN with a mitochondrial localization sequence, so you can deliver the TALENs into the to the mitochondria very easily. So in a collaborative project with a professor at the Salk Institute, we actually designed a few TALENs with increased specificity towards a gene that is associated with mitochondrial disease. And then they evaluated our engineered TALENs in a mouse model and showed that it can actually cure that mitochondrial disease in the mouse. And then in addition to the protein engineering, we also try to leverage this platform for pathway engineering. So in a collaborative project with ADM, we actually build a library of pathways that are involved in the synthesis of an amino acid. And so we use the promoters with different strengths and also enzymes with different activity, right? And we actually build like 1,000 pathways in parallel using the robotic system. And then they actually, the scientists in ADM, they screen the library, try to find the ones that can overproduce the target compound. But this step is still, although automation helps, right? Clearly now we can do more, construct, right? Or can analyze more samples. But still, you need to do it one round by one round, right? So basically, it's still not fully kind of automated. So that's why we actually want to actually develop a fully closed design, build, test, and loop, so it can actually run by itself. And in order to do that, then we really need to incorporate machine learning tools, right? Because currently the researcher basically analyzes the data and then decide what experiments will be carried out next. But if the machine learning algorithm is used, then the machine can actually think, right? Analyze the data and decide what we should be do next. So as a kind of a proof of concept, we actually try to engineer, a pathway, biosynthetic pathway involved in lycopene production. It's relatively simple, only 3 genes involved, and we want to actually develop a fully closed the design, build, test, and learn loop that can be operated many, many runs by the robotic system, right? And I can show you the details. So this pathway is only have 3 genes, and we try to actually optimize the flux. So for each gene, we want to use promoters with different strengths. We picked the 24 promoters, and so then the total combination actually will be more than 13,000, right? If you really want to find what is the best combination of those promoters for the optimized flux. And in principle, we can actually make this 30,000 pathways using the robotic system and then analyze one by one. But that will also be costly, you think about it, right? Because you have to build so many constructs. So then we were thinking that is it possible actually to use the direct evolution kind of approach, to optimize the flux very quickly. So in order to do that, of course, we have to develop a fully automated workflow first that actually includes the construction of the plasmids, right? And also the transformation of the plasmid into the E. coli, and then also extract the product and then analyze the product. Basically, it's what the researchers do in the laboratory, right? Basically, all the experience that the graduate student does in the laboratory will be automated here, right? So then once we have this automated workflow, and then we can actually only screen a very small library, only 46 pathway variants, right? Instead of 13,000, right? So we only randomly pick a very few pathway variants, and we construct those plasmids and then transform into E. coli and then analyze the data. And then actually the machine learning model actually will basically make predictions, right? Try to identify what combinations of the promoters may actually lead to high production of lycopene. And then the good thing is that now, because it's fully automated, it can run it by itself, many runs, right? We can repeat the same workflow in the second time, or even third time, or if you want, do the fourth time or fifth time. No matter how many times you want, right? It depends on the project. And so in the first round, we kind of randomly, sampling the whole protein sequence and the whole sequence space, right? So that's why you see it's a random sequencing. And then in the second round, we already have some knowledge, right, based on the 46 pathway variants we have. And then we made a kind of biased, we built a biased library, right? And then we can repeat this cycle. And indeed, in each round, we can identify variants that show the improvement in lycopene production. Right? So after 3 rounds, we got one that actually showed a significantly improved in lycopene production. But the key point is that we only actually screened less than 1% of the possible library, right? And that actually really saved the time and the cost. You think about it, right? Instead of making all the 13,000 variants, now we just need to make only less than 150, right? Those variants, and to identify the improved mutants. And in addition, actually, we also actually used a similar kind of idea. Now, this doesn't include the machine learning yet, to actually improve the tolerance of the yeast towards the acetic acid using this automated workflow. And this, of course, is still just the beginning, right? So actually, I'm currently involved in a DOE Bioenergy Research Center in which we try to develop new capabilities or better capabilities for the Biofoundry that include better tools for screening or better new kind of machine learning tools, or just more powerful automated workflow, etc. And then we also engineer a number of those organisms. And also, as Changzhu mentioned in the beginning, we also actually I'm also very fortunate to actually get a grant from NSF to launch this AI Research Institute about a year and a half ago. So in this institute, we have we try to basically develop AI and machine learning tools for synthesis planning and also catalyst optimization. And then we want to leverage those tools for synthesis of a few kind of drugs, important chemicals, and discover organic photovoltaic materials, right? And in this institute, we really try to leverage the automated system we have for biological system, but also we develop a similar system for chemical synthesis as well. And then in addition, we try to develop all the AI tools. So in the institute, half of the PIs are all actually our CS faculty. And indeed, this is very interdisciplinary. And so recently, we did develop actually a new machine learning model for protein engineering. Because of time limitation, I will not go to details. Essentially, we actually analyzed all the known proteins. There are more than 230 milling, those known proteins sequences, try to identify what, those residues may be important for protein structure or function. And then we also analyze the homologous sequence to the target protein, right, to identify those coupled residues that are important for protein function. And then we also have some mutant proteins from the target protein. Then basically we combine the data from all these 3 different sources and fed them into a deep learning model, try to make predictions. So in summary, what I showed today that, I hope that I have convinced you that directed evolution is a really powerful tool for engineering of biological system, ranging from proteins, pathways to genomes. And in my lab, we developed a number of direct evolution tools, such as the in vitro coevolution method I talked about, or the change method, but there are many other methods that we developed that I didn't have time to talk about. And then lastly, I think the most important message I want to convey is really I would say the integration of synthetic biology tools with the machine learning and automation really represents a powerful paradigm for research. So this is basically a data-driven kind of research paradigm, right? And the idea is actually kind of the direct evolution approach, because you always rely on the diversity creation, right? Or at least you create many possible constructs, and then you actually try to find the ideal, the target, desired product, but then you also have to do it iteratively, right? So then I want to come back to the question I had in the beginning, right? So what is really the answer to this global regulator? So I would argue that it really is the passion for science, right? So that makes the huge difference. So then actually I kind of applied direct evolution to create new professors in my own lab as well. As Changzhu mentioned in the beginning, indeed I have more than 30 former graduate students and postdocs who are now professors around the world, right? But I think they are all passionate about research, and they actually now they try to use the similar approach to their students as well. So finally, I would like to thank all the students who did the work I talked about, and I did mention all their names. And particularly, I want to acknowledge the funding from DOE. So most of the funding I have in my laboratory actually from DOE, at least are related to the development of the Biofoundry, right? And then also NSF for the development of AI machine learning tools. So thank you for attention, and I will be happy to answer questions.
Thank you very much, Huimin, for the very nice talk, and it's very inspiring for us to the idea how to direct evolution of not only the biology but also to become a professor. Yes. So now
not just professor, actually, so any
any kind
research.
Of,
yeah, exactly.
The development. Yeah. Okay. So now the questions.
Thank you for the interesting talk.
Can I hear very well?
So,
yeah, thank you.
Recently, we had the development of an AI technology, the AlphaFold, and I was wondering whether this influences your research as well, or whether you envisage the development of similar tools for the type of research that you are doing.
Yeah, that's a very good question. I mean, our current machine learning model actually is mainly based on sequence, right? But we also want to include actually structural information. And then the AlphaFold really provides a very powerful tool for us to basically get the structural information for the targeted proteins. So right now we are actually using AlphaFold and to build many actually structural models for the proteins that we're working on, just for direct evolution studies, but also for machine learning studies. I think that also will be useful as well.
Thanks for a very nice talk. I have a related question on protein structure.
Yeah.
So based on your experience, when you study all the mutations that are successful
Mhm.
and in proteins, and you map those mutations back to the protein 3D structure or linear
Yeah.
structure. Do you see some general trends whether these mutation steps give you what you want? Are they in the active sites, or are they in the interaction surfaces of proteins, or are they more randomly
Yeah.
distributed?
Actually, in many direct evolution studies, the mutations that were found, they are all actually outside the active site. But in the example I showed today, right, because we only try to improve the selectivity, then we actually typically target the active site residue. But if you want to just improve the catalytic efficiency, thermal stability, or tolerance to low pH or whatever the other functions, then the mutations you find, I would say, in many cases are outside the active site. And those are very hard to predict. That's actually one of the rationale why we have to use the direct evolution approaches, or at least the one strong motivation.
Thank you. Is there any more question? So
Maybe
actually,
my talk is very clear.
Yes,
That's good.
yeah. So actually, I have a question. You make the laboratory become automatically with the machines. So do you have ideas for the future? How can we educate the PhDs if all the lab is empty without humans?
No, I don't think that. Well, I mean, the students can actually be involved in the design of the experience, right, and also learn, from the experience, right. But they can, at least the robotic system will free students from the kind of routine or tedious work, like doing the pipetting. You think about it when we make those constructs, right? If you want to do it manually, it involves probably millions of pipettes, right? And then who want to do it, right? But with the robotic system, you can do all these things. Yeah. So I think that's probably the advantage. But also, I don't think for the students in the audience, you need to worry that you will lose your job, because you have other challenges, other things to do, right? You can become expert in like machine learning, or expert in operating the robotic system, or expert in designing, the new strategies, right, for engineers. You have a lot of things to do. Because I remember when I was a graduate student, I actually had to do DNA sequencing by myself, right? That was in 1992, right? So I have to pull the gel myself and then run it, and then you also use the P32 as a label, right? And then you can, and then put it actually on the like a whiteboard, and then you actually visually check, right, the band and then read out the sequence, and then you only can read out probably no more than 300 nucleotide, right? And then a few years later, of course, now we can send the DNA to the sequencing facility for sequencing. And nowadays, even we just send the DNA to a company that they can do all the sequencing, right? But once we know all the sequences, we actually have more questions, right? And we have more things to do, right? But we just didn't do the actual sequencing anymore. So that's, I think, will happen here as well. Maybe you don't need to do a cloning anymore. You maybe don't need to make the plasmid construction, but you still have to do a lot of analysis, and computational studies, or other experimental studies.
Yes.
I have another question. It has to do with exploration-exploitation dilemma. So in some of your studies, you seem to explore the space and then gradually converge to exploitation.
Yeah.
And I was wondering, so how do you represent uncertainty and whether you represent it? So one thing that you need to do that, I guess, is to represent the quality of the different alternatives, and then you learn it over time. But now many of the algorithms that are used in machine learning also represent uncertainty, and then they use this uncertainty in order to explore the space efficiently. Is this something that...
Yeah, that's actually what we're doing actually in the AI Institute. That's a very good question, is how do we estimate the uncertainty? In the example I just showed, that is actually kind of rudimentary, I would say, because we just want to show that we can have a closed design, build, test, learn loop, because that has never been demonstrated before, right? So we just want to show that it is possible to close this loop by incorporating machine learning. So the machine learning tool we use is, if you're expert in this area, is really the basic stuff. It's a Bayesian optimization coupled with Gaussian, distribution. Right? It's the standard tool. It's not fancy at all, right? But we just want to show this loop can be made. But now, indeed, like when we try to develop a self-driving Biofoundry, we actually we are trying to develop more and more sophisticated machine learning models. We try to use those models to predict those gene targets, but also actually analyze the limited number of those variants and try to make predictions as well. And then clearly we have to consider the uncertainty, because the number of variants we have is very small. Because no matter how powerful the robotic system or the genetic tools are, we still cannot make thousands or tens of thousands of variants, right? Because it's just too costly, because it also requires a lot of materials, right? And other things, it's not just the time, So we still can only make a limited number of variants and analyze them. And then I think the uncertainty analysis actually is very important. And also we try to leverage as much as possible all the known knowledge in the literature about metabolic engineering. So we could incorporate a transfer learning kind of tools, right?
So I don't see the first questions. It's pretty much interesting, inspiring talk here. So let's give the hand to Huimin. Yeah, thank you very much.
Okay, thanks.